Hi there, and welcome back to our second video on the Great Oxidation Event, where we're going to focus on the causes of this significant change to Earth's atmosphere and environment. So a first recap of the timing. So the main increase in oxygen occurred between about 2.45 and 2.3 billion years ago. After it, oxygen levels weren't the same as they are today. They were perhaps only about 1 to 10% of our modern levels, but that's still enough for tremendous change. So this general timing is fairly well established now, but there still are a few points of debate. First, were there short pulses, or whiffs as they're often called, of oxygen prior to the main event? And also, was there perhaps an oxygen crash about 1.8 to 1.9 billion years ago after the main event? Well, we're not going to focus on those today, but we're going to talk about a debate over what caused the Great Oxidation Event. Well, the source of the oxygen itself isn't really a matter of debate. It almost certainly was produced by a process called oxygenic photosynthesis and by cyanobacteria. So oxygenic, which just means oxygen producing, photosynthesis is the process of using the energy from sunlight to split water and then using that energy to convert inorganic carbon into organic matter. It's a sophisticated process that produces way more energy than other sources, so it would have been a big game changer for life. But you might be thinking, if basically everyone agrees that photosynthesis produced the oxygen, where's the debate? Well, there's two main hypotheses for how cyanobacteria may have caused the Great Oxidation Event. One is that the group evolved right around 2.45 billion years ago, which immediately led to oxygenation of the atmosphere. I should say immediately in a geological sense, which might be millions of years. The second idea is that cyanobacteria evolved earlier, perhaps even hundreds of millions of years earlier, but that the oxygen produced by them didn't build up in the atmosphere for some reason. The second one certainly does sound less likely, but it's important to see what the evidence better supports. And one way that we can do that is to look at the evidence for when cyanobacteria first evolved. So let's do that. So there are widely accepted stromatolites as old as about 3.45 billion years. Could they be a cyanobacteria? Well, on the one hand, the shape of some of these stromatolites is consistent with photosynthetic bacteria, growing preferentially in certain directions due to the brightness of sunlight. But there are other types of photosynthesis that don't generate oxygen. So this might not indicate cyanobacteria or oxygenic photosynthesis. So in this photo, the curved layers, the darker layers, are called microbial mats. They're basically layers of organic matter created by groups of bacteria living on the seafloor about 3.4 billion years ago here. Well, because these mats are found in shallow water with sunlight, but not in darker, deep water, that's pretty strong evidence for photosynthesis producing abundant organic matter in shallow habitats only. But on the other hand, the other aspects of the rock chemistry at these sites suggest that no oxygen was being produced, so this wasn't oxygenic photosynthesis after all. We can also look at carbon isotopes, which you've already heard about. And you've already heard that they have a characteristic negative shift, or fractionation, in organic matter throughout Earth history, both before the Great Oxidation Event in the Archean and after it. So it is true that photosynthesis done by cyanobacteria produces this type of isotope fractionation. So that would be consistent with cyanobacteria occurring well before the Great Oxidation Event. But it's also true that other photosynthetic bacteria and even non-photosynthetic bacteria, which get their energy from chemical reactions, can also produce and do also produce similar isotope fractionation. So this might not tell us much about the mechanism after all. Well, actual microscopic fossils would really be the strongest evidence for cyanobacteria. And you've heard about the apex chert microfossils in Australia that are about 3.46 billion years old, but which are now really quite heavily debated whether they are even fossils or not. But regardless, they're generally no longer considered to be cyanobacteria, even if they do end up being other types of bacteria. So the oldest sort of fairly well accepted cyanobacteria fossils are about 2 billion years old or so. And you might spot a seemingly obvious problem with this. These fossils occur after the Great Oxidation Event completely, and so that isn't consistent with either of our two hypotheses we're trying to test. So what does this mean? Well, it turns out that fossils will typically underestimate the true time of origin, especially when those fossils are rare. You might think, what are the chances that these cyanobacteria in the pictures here are really the first ones that ever evolved anywhere on the Earth, given how rare it is to fossilize cyanobacteria? It's almost certain that cyanobacteria evolved before these fossils were found, or before these fossils were formed, I should say, but we just haven't found any of them yet, or maybe they don't even exist, maybe they didn't fossilize. So it doesn't really help us to know the exact time cyanobacteria first evolved, other than it's 2 billion years or older. Well, rather than the whole fossil, perhaps we could look for specific chemical molecules that are produced just by that type of organism, or that group of organisms. Scientists call these biomarkers, and one type of biomarker that's pretty common are the molecules that make up the walls of bacterial cells or other cells. In this picture, these are the little balls and strings called phospholipids and the little chains called hopanoids. You might think, like, well, why don't we just use DNA as a biomarker, like in crime shows? You know, clearly DNA would be the most distinctive biomarker. You could tell exactly what type of organism you have. But unfortunately, DNA is a very fragile molecule, and it does not survive over billions of years that we would need here. So the scientists need to balance a molecule being fairly distinctive with a molecule also being fairly robust and preserved well. So biomarkers are analyzed with these sorts of spiky graphs here, where each peak is a different molecule, from the smaller ones on the left-hand side as they come out of the machine to the larger ones on the right-hand side as they come out of the machine. And so one type of molecule with the technical name 2-alpha-methylhopane, that's not super important, the name, was found in small amounts in oil in 2.7 billion year old rocks. Those are the two little peaks in the inset with the red arrows there. So if that's true, this would be strong evidence that cyanobacteria actually did evolve several 100 million years before the Great Oxidation Event. However, scientists later decided that the oil actually seeped into the rock some other time, perhaps after 2.2 billion years ago. So that's younger than the Great Oxidation Event. Doesn't really help with either of our hypotheses here. It's also possible that this particular biomarker isn't exclusively found in cyanobacteria, as once was thought, perhaps can be made by other groups as well, although maybe not as commonly as it's made by a cyanobacteria. So, yet another way we can look at this is to try to estimate the timing of evolutionary events using something called the molecular clock. So, an extremely simple way of thinking about it uses the rate of change in DNA or some similar molecule during evolution to try and calculate back and estimate when an ancestor evolved. That rate is calibrated from known fossils, and then we sort of extrapolate back to estimate the age of some less well-known ancestor that lived longer ago. So to give a very, very oversimplified analogy here, imagine we have a bunch of people who all share a great-great-great-grandparents who lived a while ago. And we want to know when that great-great-great-grandparents lived. Well, let's say we have a few known values. We can say, okay, well, in these people here, we know their parents lived were born 27 years ago. These ones we know their grandparents were born 59 years ago or 62 years ago, for example. We could then sort of use these points to calibrate, to get a scale on our branching tree here, and say, like, oh, well, maybe a generation is about 30 years. So the great-great-great-grandparents about 5 generations ago, that's maybe 150 years then if we just do the math. Now, this is obviously super simplified. This is not really what, not exactly what molecular clocks do. It's an analogy. It works differently for the evolution of species compared to your parents and your grandparents, but you hopefully get the idea at least. Here's a real example of one of these trees. Each name on the right is a different type of cyanobacteria, and the horizontal tree of branches there shows the evolutionary relationships going all the way back to the first splitting point, which is the origin of cyanobacteria. And by doing this method of sort of calibrating this tree and then calculating back, the scientists could estimate that cyanobacteria first evolved about 3 billion years ago. And multicellularity, which are sort of cyanobacteria living in small groups, which is important for producing lots of oxygen, maybe evolved about 2.7 to perhaps 2.5 billion years ago. So that supports our idea that cyanobacteria evolved perhaps a while before the Great Oxidation Event. However, there are error bars on these estimates. We say 3 billion years ago, but there is some range because it's not, there's some range of how precise this can be. It's actually possible that cyanobacteria evolved earlier, or in our case later, than these best estimates. Perhaps they did evolve right around the Great Oxidation Event. The error bars are consistent with kind of both of these ideas, although the best estimate is consistent with them evolving earlier. So let's recap all this evidence. Well, stromatolites existed long before the Great Oxidation Event, as did microbial mats produced by photosynthetic bacteria. But both of these could be formed by non-oxygen producing photosynthesis, right? There's photosynthesis, but perhaps not oxygen in this type. The best accepted cyanobacteria, microfossils, are only about 2 billion years ago, so they don't really help us know exactly when cyanobacteria evolved. The biomarkers that were found before are no longer thought to be reliable, so again, they only occur after the Great Oxidation Event and are perhaps not that helpful then. The molecular clock, could be consistent with cyanobacteria evolving as much as 3 billion years ago, although there is some uncertainty on this as well. So there's still a lot of debate over which of these two hypotheses is better supported by this evidence. What do you think? Do you think it's more likely that they evolved right around the Great Oxidation Event, or perhaps sometime earlier? So while understanding this evidence, the timing from this evidence is very important, there are also some other predictions that we can consider for both of these hypotheses. First, if cyanobacteria did evolve around 2.45 billion years ago, at the beginning of the Great Oxidation Event, what caused these so-called earlier whiffs of oxygen, 2.5, 2.7 billion years ago? Perhaps they were other rare and or non-biological ways of producing small amounts of oxygen in sort of that had local effects. Maybe the chemical evidence for these whiffs isn't a reliable signal of oxygen at all. Maybe the chemical evidence represents later alteration of the rock or some other process that we haven't considered or even thought about. But on the other hand, if cyanobacteria did evolve as much as 3 billion years ago or 2.7 billion years ago, why was there a several hundred million year long delay between their evolution and the buildup of the oxygen they're producing? Well, maybe something limited cyanobacteria, their growth and their oxygen production, such as limited nutrients or some other unfavorable conditions, and that changed around 2.4 billion years ago, which let them produce lots more oxygen. Or perhaps the oxygen produced by cyanobacteria was used up in chemical reactions with other elements, maybe iron or methane or gases from volcanoes or something else, and it took a while for those other elements to be used up, or something changed in the volcano gases or something like that, where these other elements weren't being produced as much. So overall, for questions such as these, Scientists involved in this type of study need to use multiple lines of evidence to determine which conclusion best explains all of the different pieces of the puzzle. All of these indirect lines of evidence, we have to decide, what happened billions of years ago by thinking about, well, what is the best explanation for all of these different puzzle pieces? So it can be challenging. But it's, to get at these fundamental changes back in Earth history, you have to sort of wrestle with these challenging and difficult questions. Thanks for listening.